Welcome to Developmental Biology. Today, we are going to explore one of the most fascinating processes in all of biology, gametogenesis. Imagine you are building a brand new human being from scratch. To do this, you need two very specific components. You need a tiny, highly mobile delivery vehicle carrying half the instruction manual, and a massive, nutrient-rich vault carrying the other half of the manual, plus all the building materials. The incredible, highly coordinated factory process that manufactures these two specialized cells is what we call gametogenesis. Without it, reproduction would be completely impossible. Now that we understand the basic analogy of the delivery vehicle and the vault, let us look at the formal biological definition. Gametogenesis is the biological process by which diploid precursor cells undergo cell division and differentiation to form mature haploid gametes. In males, this specific process is called spermatogenesis. It takes place in the testes and results in the formation of motile spermatozoa, or sperm. In females, the process is known as oogenesis. This takes place in the ovaries and results in the formation of a large non-motile ovum, or egg. These two distinct pathways are the foundation of sexual reproduction. To understand how these specialized cells are actually made, we must look at the core mechanism driving gametogenesis, which is a specialized type of cell division called meiosis. Unlike normal cell division, meiosis intentionally halves the genetic material. It reduces the chromosome number from a diploid state, represented mathematically as 2n, to a haploid state, represented as n. Why is this reduction so critical? It ensures that when two gametes fuse during fertilization, the original chromosome number is perfectly restored in the new embryo, maintaining the stability of the species from one generation to the next. This brings us to an important question. Why does gametogenesis matter so much in the grand scheme of biology? If our sex cells were produced through normal cell division, they would have a full set of chromosomes. When they fused, the resulting baby would have double the normal amount of DNA, which is fatal. Gametogenesis prevents this genetic overload by precisely cutting the genetic material in half before fertilization ever happens. Furthermore, during this process, chromosomes swap pieces of DNA in an event called crossing over. This creates entirely new genetic combinations, which is exactly why you look a little bit like your parents, but are still entirely unique. It is the fundamental reason populations can adapt over time. Now that we understand the biological importance of meiosis, let us look at the mathematics of cell division in gametogenesis, starting with males. The formula for sperm production is S equals P times 4. Conceptually, this comes from the fact that every single primary spermatocyte undergoes 2 rounds of meiotic division to produce exactly 4 functional sperm cells. In this equation, the variable S represents the total number of mature sperm cells produced. The variable P represents the initial number of primary spermatocytes entering meiosis. If the number of primary spermatocytes increases, the number of resulting sperm increases by a factor of 4. This massive multiplication factor highlights the incredible scale of sperm production, ensuring males can produce millions of sperm continuously. Before we move on to female gametogenesis, let us see this formula in action with a concrete example. We are given a male organism that has exactly 150 primary spermatocytes undergoing meiosis in the testes. We are asked to calculate the total number of mature spermatozoa produced. We choose our sperm production formula because it perfectly models the biological reality of spermatogenesis. We substitute the value 150 for P, representing our starting pool of cells. We multiply this number by 4 because meiosis consists of 2 distinct divisions, ultimately splitting one cell into 4 spermatids. The final calculated answer is 600 mature spermatozoa. Physically, this means the male reproductive system has successfully manufactured 600 individual delivery vehicles from just 150 precursor cells. Having seen how males produce gametes, let us now examine the mathematics of female gametogenesis. The formula for ovum production is O equals P times 1. Conceptually, oogenesis is fundamentally different from spermatogenesis because the division of cytoplasm is highly unequal. The body ensures that the single surviving egg retains almost all the cytoplasm and nutrients needed to support a developing embryo. In this equation, the variable O represents the total number of mature ova, or eggs, produced. The variable P represents the initial number of primary oocytes. If the number of primary oocytes increases, the number of eggs increases by exactly the same amount, a 1-to-1 ratio. The other potential cells do not survive, which we will calculate next. Now that we have our ovum production formula, let us apply it to a specific scenario. We are given a female organism with exactly 150 primary oocytes that are stimulated by hormones to complete oogenesis. We need to calculate the total number of mature ova produced. We use our specific oogenesis formula because it accounts for the massive unequal division of cytoplasm. We substitute the value 150 for P, representing our starting cells. We multiply this initial number by 1 instead of 4 because the egg must hoard all the cellular resources, leaving nothing for the other cells. The final answer is 150 mature ova. Physically, this shows that exactly 1 functional egg is produced from each starting cell. This unequal division we just saw leads us to an important question. What happens to the rest of the genetic material? This brings us to the formula for polar body production, which is B equals P times 3. Conceptually, since each primary oocyte undergoes meiosis to produce a total of 4 cells and only 1 becomes the ovum, the remaining 3 must become polar bodies. In this equation, the variable B represents the total number of polar bodies produced, and P is the initial number of primary oocytes. As the number of primary oocytes increases, the number of discarded polar bodies increases by a factor of 3. These polar bodies are essentially genetic trash cans that hold the discarded extra chromosomes. Let us continue with our previous example to see exactly how many polar bodies are generated as a byproduct. We are still working with the same starting pool of 150 primary oocytes. We are asked to find the total number of discarded polar bodies. We choose this specific formula to account for the 3 non-functional cells produced during the unequal meiotic divisions. We substitute 150 for P and multiply by 3. The final answer is 450 polar bodies. Physically, these 450 tiny cells lack cytoplasm and will quickly degenerate and be absorbed by the body. This perfectly demonstrates the unique female reproductive strategy of prioritizing quality over quantity by sacrificing these potential cells. Now that we have calculated the outcomes mathematically, let us look at a visual flowchart comparing spermatogenesis and oogenesis side by side. On the left, you can see the pathway for spermatogenesis. Notice how the primary spermatocyte at the top, which is diploid, divides equally during meiosis 1 and meiosis 2. This results in 4 perfectly equal haploid spermatids at the bottom, which mature into 4 sperm cells with tails. Now, look at the right side, showing oogenesis. The primary oocyte undergoes meiosis I, but the division is highly unequal, creating one large secondary oocyte and one tiny polar body. After meiosis II, we are left with one massive ovum at the bottom and 3 tiny polar bodies. This diagram beautifully illustrates how males produce 4 functional gametes per precursor, while females produce just one massive, nutrient-rich egg. We have covered a lot of ground today, so let us review the most important ideas from this topic. First, gametogenesis is the essential biological manufacturing process that creates haploid sex cells from diploid precursors through the specialized cell division of meiosis. Second, we saw the stark mathematical difference between the sexes. Males produce 4 motile sperm from every precursor cell, while females produce one massive, nutrient-rich egg and 3 discarded polar bodies. Finally, remember that this incredible process not only halves the chromosome number to prepare for fertilization, but it also shuffles the genetic deck. You now understand how every new generation is made genetically unique and capable of evolutionary adaptation. Great job following along.